Number 3, this is a bioreactor for continuous production. So it's just like our simple diagram here of growing our cells in the medium, but there's lots of other things going on that we can explain. So sterile nutrient medium, so that's just there to provide the nutrients for the cells. So provides nutrients for the cells. That's the medium bit and the nutrient bit, but sterile means it only has those cells in but doesn't have any other contamination. So sterile, so no contamination with other microbes. The impeller, it's just like a propeller, but it doesn't propel it to anything, it just stirs it. So this is a stirrer. So it just mixes the cells and the medium and all the nutrients, or else the cells would drop down to the bottom. So mixes the cells, medium and nutrients. And it does that constantly. So that your cells can always have access to all the things that they need. Oxygen concentration probe. So there's various probes here, just really to keep it at the best conditions, to make sure that cells have the correct level of O2. So for example, we might want our cells to be anaerobic or aerobic. So e.g. anaerobic or aerobic conditions. Cooling jacket. So around the outside we've got a cooling jacket, and this is because the growth of these microbes means this gets very hot. So fermentation quite often produces a lot of heat in respiration, which could actually kill the cells eventually if it gets too hot. So we need to remove this excess heat. So respiration produces heat, which could kill the cells. So the jacket removes excess heat. Sparger. This is the thing down here, so we're getting air. Air is going through the filter to remove any microbes, and then it's providing oxygen or whatever gas you need there. So this one in particular is probably an aerobic fermenter. So Sparger provides sterile air, so cells can get enough oxygen. pH probe. So another one of our probes, just to measure the pH and make sure we are working at our pH optimum. So pH probe measures pH. So that optimum conditions can be maintained. Likewise, we have got our temperature probe, so measures temperature to keep at optimum temperature. So for example, enzymes need an optimum temperature, e.g. for enzymes. We've got a facility at the top to add acids or bases. So this is in conjunction with the pH probe. So if the pH probe says it's too acidic, we'll add a base, and the other way around. So can add acids or bases to maintain optimum pH. We have got something strange called Antifoam. So Antifoam is just at the top where the gases come out. Then we get a load of bubbles, and these would perhaps get in the way of these pipes. And so we're just getting rid of this foam. So gets rid of foam that forms on the surface. That could block some of these other pipes. And then the last thing, we've got filtered waste gases. So we get lots of things like your CO2 and other waste products coming off as gases. And we need to get rid of those to maintain the optimum conditions. So gets rid of waste gases to promote optimum conditions in the fermenter. And that is your industrial bioreactor for continuous production.